Hello, cell biology fans. I've had numerous people email me or ask me or tell me that we did not finish the lecture on endocytosis, and so I'm going to do this very brief flipped lecture discussing the alternative mechanisms by which things are taken into cells. We talked at length about receptor-mediated endocytosis, discussing the three different examples, talking about the uptake of iron with the transferrin receptor, or the uptake of cholesterol using LDL, HDL using the LDL receptor, and also the uptake of epidermal growth factor receptors and their uptake into the intraluminal vesicles of multivesicular bodies. Here, what I'm really talking about are the additional ways that things can be taken into cells. We talked a little bit in class about this specialized lipid raft structure called caveolae. Caveolae, as shown in this image, are these, remember, these are these flask-like structures, and I said to you, I think of a flask as being like this, but these really look like round-bottom flasks. And these are lipid structures that, as you look on the underside of a plasma membrane, each one of these circles down here is a caveoli. I'm going to remove those. As we showed you before, this right here is a clathrin-coated pit, but these other structures are very different. You can see that in the clathrin-coated pit you have that lattice structure made by the triskelion of clathrin itself, whereas in these lipid structures there's a protein called caveolin. So all caveoli have caveolin, and what's important about these particular structures is that some viruses actually enter into cells using caveoli. So that's another mechanism of getting into cells. We don't really know a lot about the function of caveoli with respect to what they do for cells on a day-to-day basis, but they clearly wouldn't be there if there wasn't a purpose. But right at the moment, that's still under investigation. We did talk about pinocytosis. Pinocytosis, I always said to you, I think of Pinot Noir, a nice wine. Okay, that's how I remember this. Wine means that you're drinking, and so this is cell drinking. Small vesicles bud off of the plasma membrane, so all day, every day, you have small little vesicles budding off of the plasma membrane, and they're actually taking up materials from the extracellular space, and they're taking in fluid from the extracellular space. This happens constitutively. Remember what that means. Constitutively means all day, every day. There's no signal involved for this. And macrophages, which is one of the most important cells in your immune system, look at this. They can take up 100% of their plasma membrane, so this is plasma membrane, every half hour. That means that in order for the plasma membrane not to shrink, you have to have constitutive exocytosis occurring as well. And so the balance between exocytosis and endocytosis allows for cells to maintain their shape and their size. Another mechanism for molecules being taken up into cells is the process of phagocytosis. The prefix here, phage, actually means to eat. And so this is cell eating. The uptake of large particles, whether they be bacteria, as in this image, this is a bacterium, it can be taken up by a cell that pushes out its plasma membrane, and this is a energy-dependent process. These guys push out their plasma membrane in something called a pseudopod. Pseudo meaning, like, and pod is like a foot. So they're actually extending their feet out around this, and when they capture this, it is then internalized into a specialized endosome called a phagosome. Phagosomes follow the endocytic pathway, going from an early phagosome into a late, and at that point it's then called an endosome, late endosome, lysosome structure. And this is the way that your white blood cells, in particular macrophage, neutrophils, and dendritic cells, these are called professional phagocytes, they take up extracellular bacteria as a means of protecting you from them. This is a really nice video from the 1950s, and partially I love this because it's an old video and it's just so simple. What you see here is a slide where they put some blood cells. These funky looking cells are the blood cells. And what happens here is, let me erase that. What you're going to see is this white blood cell. This is the white blood cell. It's actually a neutrophil. It is going to be chasing this little tiny bacteria. The bacteria is moving by random motion called Brownian motion. So random motion called Brownian motion. And what happens is this neutrophil is not moving randomly. It is actually chasing down that bacteria, and it's following something that the bacteria is shedding. And the bacteria sheds this molecule. Whoops. Let me go back and erase. So I'd like to get rid of these so you can actually watch what's going on. Okay, the bacteria moves randomly, but the neutrophil itself is going to chase a molecule that's being shed. And this is being done by a process called chemotaxis. Chemotaxis is a process that the phagocytic cells use to follow a chemical signal. The chemical signal that most of these bacteria leave is a molecule called LPS. So LPS is shed by gram-negative bacteria. Professional phagocytic cells can chase down that molecule and end up engulfing this bacteria by a process called phagocytosis. So watch this video. So you can see that the neutrophil is actually pushing the red cells out of the way. There's multiple bacteria here, but for whatever reason, this neutrophil is chasing down this one. It's pushing out these pseudopods, and it's pulling the rest of the cell behind it, and eventually it will catch up with that bacteria, engulf it, take it into a phagosome, and that bacteria will be destroyed by the hydrolytic enzymes found in that particular cell's lysosomes, late endosomes, and phagosomes. One additional thing that we talked about briefly in class was the process of taking things into cells that are polarized. Remember, polarized cells have two sides. They have an apical side. Oh, that's really fat. Let's make it a little smaller. Okay, so they have an apical side here, and then they have their basal lateral sides, which are separated by a tight junction. Remember, this is like a fence that keeps molecules that are supposed to be on the apical side, molecules that are supposed to be on the basolateral sides, on the basolateral sides of these membranes. But we can have a process called transcytosis occur from one side of this membrane to, the other side. That is called transcytosis. You can also go from the basolateral side to the apical side. And I'm showing this from the basal, but it could also be from the lateral side. And what happens in these particular cells is they have two sets of early endosomes. You can have an apical early endosome, and you can have a basolateral early endosome. Are they showing you a basolateral? Yes, basolateral early endosomes. And where molecules meet is actually at the late endosome in these particular cells. So polarized cells use the process of transcytosis to take things in and maybe transport them across the cell to other sides. And we talked about the case of maternal antibodies, also referred to as IgG. So in a mom that has a baby, they can pass these antibodies across the placenta from the mom side of the bloodstream to the fetal side of the bloodstream, and that's how babies are protected early in life. once the baby's born, it starts to make its own antibodies, but early, before it really has time to do that, it's the mom's antibodies that protect it. So this was just a last slide showing you the last little bit, right? So we talked about receptor-mediated endocytosis, mostly clathrin-mediated endocytosis. We did not talk about non-clathrin-mediated endocytosis, but there are events that do occur without clathrin. I just talked to you about caveolae and its importance in viruses. Phagocytosis by professional phagocytes, cells that eat. And pinocytosis is cell drinking. So that finishes up this lecture for those of you who wanted a final little bit here for your notes.